once Kaitlyn's screen is up. Okay, great. Okay, wonderful. So yeah, we're delighted to welcome Kaitlyn here today as our speaker for our second reproducibility session. Kaitlyn's a PhD student at the Camarades group here in Edinburgh, and she's going to give a talk today entitled Selfish Reasons to Work Reproducibly. So I'll hand it over to Kaitlyn and she can take it from there.
Thanks, Niamh. So I think we already know a little bit about reproducibility already, given that we're all part of this group. And we know that it's good from a moral standpoint, for science, for progress, but we also need to acknowledge that as academics, we're all under a lot of pressure. And working reproducibly, it does take time. And I think to really make the shift as a culture, we also need to think about the more selfish reasons to work reproducibly and how it can benefit us as well, which is what I'm going to talk about today. Just a quick introduction to me and the sort of tools that I use. I'm a final year PhD student in the Camarades group. I work on systematic reviews of the preclinical literature. So at the moment, I'm doing a systematic review on in vivo animal models of Alzheimer's disease. I also work on developing and using automation tools for systematic reviews to try and speed them up. The tools that I use day to day are things like RStudio or Markdown, which some of you may have heard of or you might use already. More recently, I've started to use GitHub. I was pretty scared to use it initially, but in the past year, I've really sort of embraced it and I use it all the time. And I also use the Open Science Framework to manage my projects day to day. I really like the Open Science Framework because you can put documents on there, you can put PDFs, all of the materials relating to your project, you can put in one centralized location. So that tends to be how I share my data. And finally, I just want to say that I'm very much still learning. These are just the tools that I use day to day, and these are the tools that I'll sort of bring into the talk. They might not apply to everyone, and I know there's so many other tools that we can talk about in future sessions. So yeah, I'm not an expert, but these are just the tools that I'm using, and hopefully I can share how I've been using them with you. So this session really came about because I had suggested this paper, 5 Selfish Reasons to Work Reproducibly by Florian Markowetz. So this came to my attention a few years ago because I actually saw a talk from Florian Markowetz at a reproducibility workshop, and it really struck me because I think until that point I'd always thought about reproducibility as kind of just from a moral perspective, it's clearly what we all should be doing. I hadn't really thought about, oh, it can also be good for me personally or my career as well. So I think if you haven't had the chance to read the paper, I really recommend reading it. It's written in very plain language. It's really easy to read, even if you don't know a lot of the terminology. And it's got plenty of examples from Florian's own work in genomics research and cancer research. So I think it's a really good read. And one of the best quotes is at the start of the article, sort of repurposed quote. So my fellow scientists, ask not what you can do for reproducibility, but what reproducibility can do for you. So with that, I just wanted to quickly define what I mean by reproducibility, because there's a lot of different terminology, and to be honest, not everyone even agrees on these definitions, but just for the benefit of this talk, hopefully this will make sense. When I talk about reproducible research, I'm talking about research in which the authors provide all the necessary data, materials, and the code to run the analysis again and recreate the results exactly. Replication, on the other hand, is more when you repeat a study. So you repeat the same methods, but you maybe do it on a different population, you collect different data, and you do a different analysis. So although you're trying to replicate some result, it's in a different situation. There's a lot more caveats to this. There's a lot more things that can change with a replication study. So that's not what we're talking about today. We're just talking about reproducing a study in its entirety, so that so the results are exactly the same. So the first reason that working reproducibly can benefit you is that it can help you avoid major disaster. So this is an example that Florian uses in his paper as well. This was an example of a high impact paper published from Duke University. It was all about a new genomic technique they discovered to guide the use of cancer therapeutics. So it was basically that they could take a tumour, sequence it, and say, you know, which chemotherapy would target that tumour best. So as you can imagine, this was a really big deal at the time. It came out in Nature Medicine. There was a lot of hype about it. Cancer clinical trials were actually started based on the findings of this study, and researchers around the world were trying to replicate the findings or reproduce the results. So at the University of Texas, Keith Baggerly and Kevin Coombs are statisticians there, and they tried to actually reproduce the results. So they went through the paper and, you know, looked at all the statistical techniques and really tried to get the same results. There was a lot of back and forward with the original authors who weren't that forthcoming about, you know, exactly the methods they'd used and weren't really sharing their whole data or their whole code. It was a bit unclear. But after an amount of time, Baggerly and Coombs actually realised there must be something actually wrong with the data and tried to have a sort of forensic approach to look back and say what must have gone wrong to arrive at this data set or to arrive at these results. Some of the things they spotted you can't fully see in this slide, so I've kind of added over the slide so you can see. They had a data set of genes, which is what all these things are. They're all sort of numbers with letters, which don't really mean anything to me, but they're all different genes basically. And in the original data set, it had 1881AT. On the same row in Baggerly and Coombs's data set, they had 188AT. So they started to realise there was some systematic error in the way that the original authors had run their analysis. And it turned out that the original authors had been using Excel. And they'd copy pasted rows from Excel into another statistical program, and it had caused some off by one error where everything had sort of increased by a number, and it actually rendered their whole analysis completely false basically because all the genes were wrong. You can see how this mistake could have happened. It's not inconceivable. However, in this paper, there were a number of other errors as well, inconsistent labelling, duplicated samples, and in the end, it actually amounted to scientific fraud. So although a lot of the authors on this original paper didn't even take the time to look through the analysis properly and see these issues, it was really damaging to all of their careers, and it ended some of their careers as well when this paper was retracted. So this is kind of like worst case scenario. Another slightly not as bad disaster happened in psychology, but it's very different. So this is Julia Strand, who's a psychology researcher who published recently the biggest finding of her career, and then a few months later discovered that actually there was a major flaw in it. Due to a programming bug, she had started the timer in the control group a little early, so it meant that the control group looked slower than the experimental group. It's a really easy mistake to make. The difference here is that all of her code was openly available. She was fully transparent. She'd pre-registered her analysis. Everything was out in the open. And she also spotted her own mistake and went to the journal editors. And I guess this is quite a brave decision. I think she did have a bit of, you know, should I do this? Should I not do this? But she did it. And it paid off because there was no suggestion she'd committed any fraud, because it was very clear that because everything was out in the open, it provided a safety net for her and her career was not ruined. So even in these disaster situations, sometimes it can be pulled back by you working reproducibly. Another type of disaster that you definitely want to avoid is this now infamous example from Cambridge University, where a PhD student there had all of their data, external hard drive, in their backpack and then lost their backpack. So all of that work was then gone. So you really want to avoid losing track of your data and avoid this type of disaster as well. So in summary, Working reproducibly, it does help you avoid losing track of your data, hopefully, avoids big mistakes. It can act as a safety net in those disaster situations. And overall, it gives you more trust in yourself and in your co-author's work. The second reason that working reproducibly is useful for you is that it makes writing easier. So I just wanted to show this quick video, which is essentially a horror film all about non-reproducible answers. And maybe you'll see your own work in here. Probably maybe you see yourself as the first half of that video. I'm not sure where you are on that that sort of workflow, but I've definitely been in the situation in the first half where you're ready to write up your results for your publication and everything is in different folders. It may or may not be named in the best way. You have to somehow pick out all the best parts. So your analysis file, your data file, your maybe you've got plots somewhere that you've ran in SPSS and exported. You need to pull all that together and get it into a publication somehow. So there's a lot of opportunities for error in this process, and the reality is also that you probably will have to do it again at some point. You're going to find some mistake or someone will discover something, you'll have to redo it. And the idea of having to redo all those clicks in Excel or SPSS exactly the same way is not a nice prospect. So there is another way. As you saw, that little video showed a workflow in R, which I'll sort of talk about here as well. If you use R, the first step to becoming more reproducible is to use R projects. So this is a basic R project setup. It's just something you can do when you open up R. You can just create a new project. And the idea of it is that you have all of your scripts, all of your data, all of your outputs in one folder, and you can refer to it really easily within R by just saying, you know, raw data, my file, and then data, this file. You can just keep everything in one place, and it just makes everything really tidy and easy to share later. So this is one example from my own work where, yeah, you can see I kind of have it in this format, and I've got raw data, and I'm bringing in this file, question ID, checklist ID, CSV. So once I'd created all this stuff in RStudio on my computer, I wanted to make it open, so I put it on the Open Science Framework in exactly the same way. So I basically mirrored the R project on the Open Science Framework, so you can see this is the raw data folder on there, and I've got the same file here as well. And what this means is, because it's structured in this way, someone should be able to download all of my folders on here and rerun the analysis if they want to, which is exactly what we're talking about. If you want to go a step further, you can use what we call literate programming. So this is a methodology that combines a programming language with a documentation language. So when I wrote my R script for analysis in this project, I wrote it in R Markdown format. So I think some people think this is really, really different from R, but it's just written in pretty much the same way. It's not a big difference. And when I was doing this, I didn't have that much experience with R, so I don't think this is something you can't do if you're quite new to R. So you can see this is my script here. The main difference in R Markdown is you can see these grey boxes. These are kind of code chunks, and it just means that you can click this little green triangle here and you can run the whole chunk in one go. But the main advantage to using R Markdown is that you have this option up here, you may or may not be able to see, it says knit. You can press this button, and using the knitter package, you can convert your script into a HTML document, a PDF, a Word document, and lots of other formats as well. So this is the same script in HTML format. You can do lots of fancy things as well. You can add in nice graphics and lots of things. I added in just this little drop-down scroll box where you can click through my main variables, set up all the different analyses that I did. So you can see already this is a much nicer output. It makes it easier for me to look back on and for people to, for your collaborators to read. And because it's literate programming, you can put lots of text in there as well to accompany the code and really put as many comments in as you want to document it. So at this stage, I still wasn't super comfortable with R, so I still ended up doing a little bit of copy pasting. I basically copied this graph into the final paper. Nowadays, I'm ready to probably take things a step further again, which is that you can write the entire paper in R Markdown. So this is an example of a paper it's a paper about writing a paper in R Markdown. And you can see it's got, it's really nicely formatted. It's got an abstract section here. It's got footnotes. You can put graphs in. You don't even have to leave all the code in if you don't want to. There's options to, hide the bits of code and just output the graphs or output text. That's totally fine. This articles package within R actually lets you export to different formats for different journals. So you can see some of the journals here. You've got Frontiers, the archive journals. You can also do PLOS, and there's more journals being added all the time. So it means that you could theoretically write your whole paper, know exactly the code behind every figure and how you arrived at every result, and just export it in that way and submit to a journal. If you want to go further again, at the point where I did this work, I wasn't really collaborating with loads of people on the code, but nowadays I am, and we use GitHub now. So GitHub, for those that don't know, it's basically a versioning system for code, and it really helps you keep track of the history of your project throughout its life cycle. So you start off with a bit of code on your computer, you basically push it to a remote repository on GitHub online. The code goes on there, and then people can change that code. They can pull it into their own computer, they can change it, and then they can push it back to the remote repository, and then you can pull back everything, pull back all those changes into your computer, and you have the most up-to-date version of the code. So that's kind of what that video was showing at the end there. That's the perfect scenario where your collaborator changes a bit of code, you pull it all into your computer, and then you knit it. It's a PDF, and that's your paper. So that's a kind of ideal workflow that's fully reproducible and saves you time in the long run. So reproducible workflows overall, they're easier to update when data changes, they're easier for people to read and understand, and they're easy to convert into paper format. The third reason that reproducibility benefits you is that it can ensure the continuity of your work. So this is a brilliant quote, which definitely rings true. Your primary collaborator is yourself 6 months from now, and your past self doesn't answer emails. I've definitely been in this situation, my past self for not leaving enough information and notes. I think when you're really engaged in a project and you're kind of racing to get to the end, you just think that you'll just sort it out at the end, and you'll just share all the stuff at the end, and you'll just remember what you did. But the reality is you really, really won't remember what you did. And all those little decisions and those little tweaks that you made, you really won't remember. So with programming, again, you can add in a lot of description that will help you later on. So this was again from a previous project, and the sort of detail that I include is things like what all the variables are. What do they all mean? And I've also left some notes for myself to say this response is different for this reason, and this is why this is different. Because I know when I go back to the code, if I try and work through it again, I'll come up against the same problems and I'll be like, oh, I've made a mistake. Why did I do it this way? Why did I do it that way? So many notes for yourself as possible. This code on the Open Science Framework has been downloaded 28 times, and I'm pretty sure at least 26 of those times have been me downloading it again. Because I documented it in this way, it's super useful because I can easily go back and see what functions I used back then, and I understand what I did. So it's really, really beneficial. Thinking more as a lab perspective, if you work collaboratively with a lot of people, a question that was often asked in our lab is who has the code for running that analysis? And we all had bits of code on our computers, sort of hidden away in different folders, and it was really a bit of a nightmare to try and coordinate. So now finally we have a Cameradi's GitHub page, and we all put our code on there so that as a lab we can share and reuse that code. For anyone that is not aware of this, GitHub now have allowed you to create a private repository within a group. So it means that your lab could have a group and put private repositories on there that only you could see. Obviously, we're talking about being open here, so ideally you do want to make things public, but there's a number of reasons why you can't do that, and that shouldn't stop you using GitHub if you do need to have a private repository. Also, just a note to say that just by putting all the codes on there, obviously it's not immediately useful. You also need to have README files, which in our case, we're trying to write README files that say this code specifically does these things. You need to be really explicit. And this means that people even that don't write code should hopefully be able to open your code and run it and understand what it does. This was another common question in our lab, which was, can you show this new lab member how to do this? So I wanted to show this example from our sister organisation in Berlin, Cameradi's Berlin, and they've created this amazing systematic review handbook. This was created using Bookdown, which is very similar to R Markdown, except that it just binds all the R Markdown files together into a book format. So this can be really useful if you want to have a sort of lab handbook. You can put code in there, you can put lots of text in there. It's really easy to update. You can link it with GitHub and everyone can make changes and collaborate. But this is something that's worked out really well now, because now they can just direct new students to this resource rather than having to explain it themselves. Something we've used in Edinburgh recently is R notebooks, which differ slightly from R Markdown in the R Markdown basically runs everything. It runs all of your code and then it outputs into a PDF or whatever. And R notebook is more meant to be used when maybe you've got a student who will be running a really similar analysis to you, but they're changing little bits of the code. So you can get it so that you can preview in a Markdown format, so it will appear like this here. And then you can actually change the code interactively and follow along with this looking HTML page here. And it's just a much nicer way to work, especially for people who are not that confident with code yet. So overall, this means that you and others can build upon your previous work. You're not going to repeat yourself to the same extent, and you can prevent situations like this cartoon here, where someone comes into the lab and has to try and work out what a previous person did. And a lot of that time, that work can actually be lost because it's not been documented well enough for someone else to use it. Another reason that working reproducibly can help you is that it can help you get your paper through peer review. So it helps reviewers understand what you've done, especially if you're using some sort of programming like R Markdown or similar. Florian actually gave an example in his paper that a reviewer had downloaded all of his materials and data and rerun his analysis. I'm not sure how common that is, but I think as things progress and we use more of these literate programming things where you can just click a button and rerun the analysis, I think it will become more common, and hopefully it will get everyone on the same page and to help reviewers actually understand what you've done and open up real discussions about it. And it also enables you and your co-workers to make the changes that the reviewers suggest without getting as angry, because it'll be easier to change. Another reason working reproducibly benefits you is that it can future-proof your work and it can help build your reputation as well. So we all are probably aware that increasingly stakeholders are setting out requirements for data and code sharing practices. Maybe it's not happening fast enough, but the revolution is here, it is moving. There's now funding being specifically offered for replication projects and open science projects, and eventually it does seem like reproducible science culture will become the norm. So early adoption will hopefully confer a future advantage for people that do that. We can also see this reflected in new publication formats. For example, over at eLife, they already have executable research articles, which are basically when you have the figures in the article, you can click a little drop-down button and you can see the code behind those figures. So I think this is a really exciting development, and I think more and more journals will adopt these sort of practices in the future. Over at F1000, they have living articles and living figures. So this is where the text and the figures can actually update based on more recent information. So this is great for things like systematic reviews where maybe more data will come in over time. This can update in line with that. A bit like an R Markdown file, it's really easy to update once the data changes. You can also now have LaTeX submissions to journals. So I haven't really spoke about LaTeX, but essentially the way that knitter package and R Markdown and articles work is that it converts the scripts into text format, and one of those text formats is LaTeX. So LaTeX, sorry, always say this wrong, LaTeX. And now you can submit just that. So basically it means that you can take that R Markdown document and submit directly to PLOS One, for example, that would just be great. There's also now data descriptor articles. I've put a couple of examples of journals here. BMJ Open Science offer them, so does Scientific Data. And this allows you to basically have your data as another publication, and people can then cite that data, and you go into detail in that publication about how you generated your data, what all the columns mean, and really describe your data so that it's usable for other people. So finally, I just wanted to say something about open science as well, because you can work reproducibly in a way without being fully open. So you can sort of work reproducibly within your lab and you can know that you can reproduce your own results. But open science has a benefit as well that I just wanted to talk about. So if you have a transparent workflow, you have open data, reproducible results, you publish in an open access journal, maybe you use a preprint as well, maybe you publish in a preprint server before your full results are out there, it's going to lead to increased awareness and accessibility of your work. It means it's more likely that other people are going to be able to reuse your data or your workflow or your functions that you've created. It means that there's going to be increased discoverability for your work. And overall, this leads to more citations of your work, your data, your code, more recognition, and more opportunities for collaboration. So overall, it gets your work noticed, and it does benefit you in that way as well. I just wanted to end on my own selfish reason as well, just since I've been given this platform, so I just wanted to mention this as well. As a meta researcher, our main sort of goal, or my main goal, is trying to get data from publications. And at the moment, we've got a crowd of reviewers working on a systematic review, and sometimes they can't work out how to get the data from the article. So at the moment, for context, we quite often have to go to the figures in that paper and literally measure with a ruler to work out what the mean is, what the error bars are, if it's not presented in a table, which is not fun. And if everyone was to make their data available, it would be much easier, because I've asked my reviewers to explain why they can't extract data from publications in some cases, and these are the sort of responses that I've got. So authors say error bars were omitted for clarity. Not sure how that helps anyone. There's mistakes in the ordering of the figures, or the axes are wrong and it's unclear what figure relates to which text. This actually happens all the time. Sometimes the figures are unreadable. Sometimes they've performed the study but the data isn't shown. All of these problems are immensely frustrating, and if people could share their data and make it obvious what they've done, that would also really help me. So that's my selfish reason. So I don't know if I've convinced you so far about the benefits of working reproducibly. I just wanted to kind of finally highlight some of the time benefits and weigh up the time it takes and the time you'll save. So by working reproducibly, there will be time spent in learning new software. Maybe you currently code, maybe you currently do your analysis in Excel or SPSS, and you will have to learn, R or Python or something, and that will take time. It will take time. You also have to spend more time planning out your experiment from the start, and you have to spend more time documenting each stage of your workflow. However, if you don't work reproducibly, you're going to spend more time compiling your analyses and your papers, especially if you have to redo them. You're going to spend more time trying to remember what you did months ago. You're going to spend more time repeating yourself to other people, and you do have a higher chance of missing big mistakes. So hopefully I have convinced you that reproducibility is good for science and is good for you. On a final note as well, I think a lot of people think reproducibility is just at the end. I'll just make all my data available now and I'll just write it up at the end. But ideally, you do want to be starting with reproducibility in mind when you start off the project. So maybe when you're formulating a hypothesis and you're designing your study, you maybe want to already think about writing a protocol and starting to think about where you can integrate reproducible tools. Maybe you're going to centralize your project on the open science framework or similar. You're going to work out a versioning system or a way of naming your files that you're going to remember where everything is. And then when you come to analyze your data, maybe you're starting to think about, even if you don't want to make the switch from Excel or SPSS to R, some of these programs like SPSS, you can download the script behind all those clicks that you're doing. So even that's a start. Just trying to introduce reproducibility at each stage of the workflow. But as this slide says, try and do what you can. And reproducibility doesn't have to be all or nothing. It's a slow process, and hopefully it will get better over time. So I just put up some final thoughts for discussion afterwards on this slide, but I just wanted to thank you all for listening.